Students, let us see now the next chapter, the cell structure and microorganisms. Now, what is the name of the minute components of which the body of a living organism is made up of? Now, we have learnt in the lower classes that the body of a living organism is made up of a cell. Now, is the number of these smallest units the same in the bodies of all living organisms? The answer will be no, because the size of the different organisms is different. Naturally, the number of cells in the bodies of the living organisms will also be different. Now, let us define the cell. Now, we have learned that cellular organization is the primary characteristic of all living organisms, and that the cell is the fundamental structural and functional unit of living organism. So, this is the definition of a cell. Now, let us see how this cell can be compared to the letters, words, sentences, paragraphs and chapters in a book. Now, all of you know that there are 26 letters in an alphabet. Now, these letters, they form the words, and the words form sentences. Sentences form paragraph. Paragraph forms the chapter, and the chapter forms the book. So, let us compare this with the concepts in the living organisms. So, we see that the structural organization of the book in the above flow chart. Similarly, there are organizational levels in the living organisms too. They are cells. Cells forms the tissues. Tissues forms the organs. Organs forms the organ systems. Organ systems means which one? Respiratory system, circulatory system, excretory system. Then these different systems together they form the organisms. Thus, the cell forms the basis of the structure and the function of all living organisms. It is only with the help of cells that living organisms carry out their different life processes. Now, let us see in this book now, in this figure now, organization in the living organisms. Now, the cells, they form the tissue. The group of similar cells performing the same function is called as the tissue. Now, different tissues forms the organ. Different organs, like here the organ system shown is the digestive system. So, stomach is there, intestine is there, large intestine, small intestine, then liver. All these different organs, they form the organ systems. And like this digestive system, excretory system is there, circulatory system is there, nervous system is there, and all these systems, they form the body. So, thus we can compare the structural organization of the book to the structural organization of the living organisms. So, now the next question is, where do the life process of the unicellular organism take place? Now, the unicellular organisms, from the name itself you can understand. Unicellular organism, it consists of a single cell. Example is amoeba, paramecium, euglena. These are all bacteria. These are all unicellular organisms. So, all the life processes, it takes place in a single cell. They do not have a separate organ system. Now, let us see about this scientist now. In the 1965, a scientist, Robert Hooke, observed a thin section of a cork under a microscope. In this section, he saw hexagonal compartments like those of a beehive. He named those compartments as cells. So, this term is derived from the Latin word cella, which means a small room. So, in 1838, two scientists, M.J. Sheldon and Theodore Schwann, formulated a theory about the cellular structure, which stated all the living organisms are made up of cells, and the cell is the fundamental component of living organism. So, in 1885, Rudolf Virchow stated that all cells are formed from pre-existing cells. Now, let us next see how the cell can be measured and observed. So in 1673, Anton van Leeuwenhoek assembled various lenses to construct a microscope. So he was the first to observe live bacterial and protozoan cells under the microscope. Now these cells are extremely minute in size. We cannot see cells with the naked eye. Micrometer and nanometer are the units which are used for measuring their sizes. The compound microscope is used for observing cells. An object on the glass slide magnified many times by the lenses of this microscope. So here this microscope you can see here compound microscope is shown in the figure. So it consists of the base, then illuminator which is the mirror, then the stage is there on which the slide which has to be observed is kept, then objective lens which is near the object is called as objective lens. Then with the help of this fine focus knob you can adjust the lenses and focus on the object. Then coarse focus knob is there, then this is the tube and eyepiece or the ocular. So through this eyepiece lens we can see the image of the object. Now here let us see this conversions here. So 1 centimeter is equal to 10 millimeters, 1 millimeter is equal to 1000 micrometer, and 1 micrometer is equal to 1000 nanometer. So this conversions means here you can see nanometer is smaller than micrometer, micrometer is smaller than millimeter, and millimeter is smaller than centimeter. Now take a piece or the segment of the onion and carefully separate the thin skin from its concave surface with the help of forceps. Place the membrane on the glass slide and put a drop of water on it. Take care that the membrane does not get folded while placing on the slide. Put a drop of dilute solution of iodine or eosine over it and observe under the 10 times objective of the compound microscope. Now 10 times, 10x means 10 times. 10 times means what? The size of the image will be 10 times the size of the object. So don't forget to put a cover slip over the onion skin on the slide before placing it under the microscope. In the same way, observe the cells from the various parts of the plants like leaves, bark, root, tips, etc. So what we see that you can observe the different cells, and after observing it is seen that all the cells are not same. Their structures are also different, their shapes are also different. So thus we see that there is a great variation in the shapes of the cells. Their shapes are mainly related to their function. Now observe the cells of different shapes shown below. Now this is amoeba, then red blood cells, surface cells, an egg cell, spirogyra, a fat cell, a muscle cell, a bone cell, a sperm, a nerve cell, etc. So these are the various cells. So these cells you can see they are of different shapes. Some are circular, rod shaped, columnar, spiral, oval, rectangular, etc. Now each cell contains various components for carrying out the life process of the living organisms. Now these components are called organelles. These organelles are studied in detail with the help of electron microscope, which can magnify images up to 2 into 10 raised to power 9, that is 2 billion times their actual size. So now you can understand electron microscope is powerful than compound microscope, and compound microscope is powerful than the simple microscope. So if you want to observe very small cells, then they should be observed under the electron microscope only. Means the size of the objects will be, the size of the image will be 2 billion times the size of the object. Now there are two main types of cells, animal cells and plant cells. Now these cells consist of various types of membrane bound cell organelles. Plant cells have a definite shape due to the presence of cell wall around them. Besides, unlike animal cells, plant cells contain single large vacuole. So all these cells, they are known as eukaryotic cells. Means again the cells, they are of two types, eukaryotic and prokaryotic. So we'll come to that point later on. Now let us first observe the plant cell and the animal cell under the electron microscope. So you can see that there is a single big vacuole in a plant cell, whereas there are many small vacuoles in an animal cell. Then there is a cell wall in a plant cell. There is no cell wall in the animal cell. Now, then there is a cell membrane in a plant cell, whereas cell membrane is the outer covering of the animal cell. Thus, the plant cell has got double covering, cell membrane as well as the cell wall. Then Golgi bodies are present in both. Free lysosomes are present in both. Lysosomes are present only in the animal cell, whereas the lysosomes are absent in the animal cell. Then chloroplast is present in a plant cell, chloroplast is absent in the animal cell. Then mitochondria is present in both the cells. Endoplasmic reticulum is also present in both the cells. Nucleus is present in both the cells. Now all these cell organelles, nucleus, Golgi bodies, endoplasmic reticulum, vacuoles, they are embedded in a fluid-like substance which is called as the cytoplasm. Now the cell has main four parts: the cell wall, the cell membrane, cytoplasm and cell organelles. Now cell wall is the outermost covering of the cell. It is present only in plant cell. The plasma membrane, also called the cell membrane, is a kind of a thin covering. It is extremely delicate and flexible. It is the outermost covering of animal cells. Then cytoplasm, it is a liquid part in the cell present around the nucleus, is called as the cytoplasm. It occupies the space between the plasma membrane and nucleus. Cell organelles are scattered in the cytoplasm. Now these cell organisms, they mainly include the nucleus, endoplasmic reticulum, Golgi bodies, lysosomes, mitochondria, vacuoles, blastocysts, etc. Now plant cells contain chloroplast. Now what are the common components of the plant and the animal cells, and which are the different ones? Now the common components from the figure itself we can see here. They are nucleus, Golgi bodies, cell membrane, vacuoles and mitochondria and endoplasmic reticulum. And the uncommon ones are which one? Lysosomes are present in animal cell, chloroplast is present in the plant cell. So these are the uncommon ones, and cell wall is present in the plant cell and cell wall is absent in the animal cell. Now nucleus is the most important organelle of the cell. There is a porous double membrane around it. The nucleus controls all functions of the cell. The endoplasmic reticulum is the sprawling net-like organelle. Its function is to make the necessary changes in the proteins produced by ribosomes and send them to the Golgi bodies. Golgi bodies are made up of several flat sacs. Their function is the proper distribution of proteins. Mitochondria and plastids are organelles with double outer covering. As mitochondria produce energy, they are called as the powerhouses of the cell. The chloroplast in plants carry out the function of photosynthesis. Vacuoles help to throw out waste products of the cell. Vacuoles in animal cells are small, whereas there is one large vacuole in a plant cell. Now the question is, how do the cells acquire definite shape? Now the cells acquire definite shape due to the cell wall and the cell membrane. Now how are the cells protected? The cells are protected by the cell wall in a plant cell and cell membrane in an animal cell. Now what are the needs of the cell? The cell needs oxygen and glucose. Oxygen is required for the respiration and glucose for the energy of the cell.